Alright, this next section is going to talk about the skin. And this is a list of the learning objectives that are in association with the integumentary system. So we'll start by talking about some of the major functions of the integumentary system. The integument is talking about the skin and all associated structures. So the integumentary system is talking about the system of skin and accessory structures all around your body. So some of the major functions include protection. There's 3 different types of barriers that the skin forms around the body. First of all, it forms this chemical barrier. Low pH secretions, known as the acid mantle, and defensins slow bacterial activity. So what we have on the surface of our skin is this chemical layer, and that chemical layer is known as the acid mantle. And the acid mantle is talking about this surface liquid. And this surface liquid environment is acidic and has a high salt concentration. There's not a lot of bacteria that can survive in the presence of acid and high salt concentrations. Have you ever seen a person that's moldy, like molds growing on the surface of their body? No, because of the acid mantle. So molds can't tolerate an environment like that. So this chemical barrier prevents the growth of a lot of microorganisms. This is not all microorganisms. There are some, like staph and strep species, that have learned and evolved to be able to live in those types of environments. The next type of barrier, it's a physical and mechanical barrier. So the keratin and glycolipids block most water and water-soluble substances, and then there's limited penetration of the skin by lipid-soluble substances, such as plant oleoresins. Plant oleoresins, that's like poison ivy and poison oak residue, so that can penetrate. But other than that, water can't just seep through our skin. That barrier prevents water from dissolving our tissues. Biological barrier. Dendritic cells, macrophages, and DNA in and of itself serves as a biological barrier. So the dendritic cells and macrophages are going to eat pathogens as they try to enter in through the skin. Macrophages are the reason why our tattoos stay in place. So when you get tattoos, they inject a dye into the dermis layer of the skin, and localized macrophages are going to eat that dye. And when a macrophage eats something that it can't destroy, it turns into this dormant, persistent form, and it stays there forever, because it's better to have trapped the foreign substance than to leave it free in the environment. So it'll trap it and then it'll just stay there, and that's why our tattoos stay in place. Now over time, as the macrophages die, neighboring macrophages will consume the dye and take over the role of what that former macrophage was doing, and the tattoo may migrate, your skin may be droopy and all that type of stuff due to old age, but macrophages, they eat stuff in the skin. And then DNA. So when UV light hits DNA, it causes the formation of pyrimidine dimers. Your pyrimidines are the bases cytosine and thymine. So anytime you have a C next to a C, a T next to a T, or a C next to a T, I'll get rid of that. It can cause them to link together. And when we have pyrimidine dimers form, as you try to do DNA replication, you're trying to read this and you see this and you don't recognize it because they're not bound like this, they're bound to each other. So this complex confuses the polymerases that are trying to do replication. It confuses the RNA polymerases that try to produce protein from this. So this can lead to a lot of errors. Now we're constantly going in and repairing these pyrimidine dimers, because otherwise every time we were exposed to UV light, we'd have some type of mutation develop. But the DNA kind of absorbs a bulk of that UV light coming in, and it alters our bases. over many, many, many years of constant exposure to UV light, there's a chance that one of these pyrimidine dimers can go unnoticed and unrepaired. And if it happens to be a mutation in a part of the cell that regulates cell growth and division, that's what can eventually lead to cancer. So UV light is harmful to the body. It gets absorbed by our DNA, and it causes these pyrimidine dimers to form. We always fix these though, so most of the time it's not a huge issue. It's just after constant exposure, when you're not protecting your DNA from this UV light, you can go on to develop some pretty serious things. So body temperature regulation. We sweat. So about 500 milliliters of water per day is lost through insensible perspiration. So we have these eccrine sweat glands that we'll learn about later on as we go through this lecture that are responsible for secreting out this water, and they're located all over the body, and this water is constantly given off, and it helps cool the body through evaporative cooling. So our body makes an assumption that the outside temperature is colder than what the inside of our body is. So if our body is 98.6 degrees Fahrenheit, how often is it hotter than 98 degrees outside? Depends on where you live. Like here, not that often, but if you go down to Texas, every day it's like 110, and it's like humid heat. It's horrible to live in. It was like torturous living down there. It's a great, beautiful place, and the people are absolutely wonderful, but the temperatures are just ridiculous, and the humidity with it just makes you want to die. So if you have humidity in the air and you're trying to sweat water is not going to want to readily leave your body because there's so much humidity in the air already. So it kind of stays on your body a little bit longer and it doesn't cool you off as well. That's why when it's more moist heat versus dry heat, in dry heat it's actually better. Like the heat in Arizona where the air is really dry helps, all that sweating helps, goes into the environment and helps cool you off. But anyways, so back to this body temperature regulation. When you throw water on the surface of your skin, that water is going to take some of your body temperature, and through evaporative cooling, it's going to take that heat away from the body. Cutaneous sensations: temperature, touch, and pain. Cutaneous is just talking about the skin. Sensations are talking about what can we feel in the skin. So temperature, touch, and pain. There's temperature, touch, and pain receptors located in the skin. Metabolic functions. So the synthesis of a vitamin D precursor and collagenases. And then there's a chemical conversion of carcinogens and some hormones that happens in the skin. It's a blood reservoir, so up to 5% of the body's blood volume can be stored in the skin. During the fight or flight response, this blood moves away from the surface of the body, and that's why some people turn pale if they're very afraid of something or if they feel like they have to fight something. So the blood will move away from the skin and toward the internal organs in order to, or predominantly the muscles, in order to provide more nutrients to them for energy production. Excretion, so nitrogenous waste and salt are excreted out in sweat. So now that we've talked about some of the major functions of the skin, we're going to talk about the regions of the skin. And the two major regions are the epidermis and dermis. The epidermis is the more superficial layer, and a majority of this is dead. The bottom part is still living, but the top part is definitely all dead. So everything that you see on the surface of a person is dead, except for the conjunctiva of the eyes. So that little membrane that covers the front part of the eye, that's still alive. The rest of everything is dead. Dermis. Dermis is the deep layer. And this is going to contain a majority of the accessory organs of the skin. So all of those glands, the hair follicle kind of arises from the dermis. Major blood vessels can go through this area, nerves, there's a lot of nerve supply, and so forth. So epidermis is the superficial layer, a majority of it's dead, and there's no blood supply. Well, the dermis is the deep layer, and it's a majority of the thickness of the skin, and many of the accessory organs associated with the skin are located in the dermis. Then you have this hypodermis, also known as the subcutaneous layer. Some textbooks say that this is part of the skin. Other textbooks say that it's not. Ours happens to say that it's not, so we're going to go with that. We're going to say it's not part of the skin. So the subcutaneous layer contains a lot of fat, and that fat is there to support the rest of the skin. So here you can see the various layers of the skin. So here's the epidermis, the dermis, and then the hypodermis is this underlying layer that follows all of the skin around the body. If you look here, all of these blue and red stringy things, those are the blood vessels, and the blood vessels stop right here. If you've ever got a blister before, with that blister, you've got a little, the skin turns white in color, and then usually there's a pocket of fluid underneath there, and you can peel off that blister and it doesn't really hurt. And there's no bleeding associated with it. Unless you like really peel it off hard and get in deeper into the skin, usually you can cut off that white region of skin and it doesn't hurt and there's no bleeding. Why is that? Well, what you're peeling off in a blister is the epidermis layer, leaving the exposed dermis. Now underneath, when you look at that, it's going to look bright red. And that's because the blood vessels are dilating under the skin because they know that the area above it has been damaged and it's going to need extra nutrients in order to repair itself. So you'll see like this really red area underneath where you've peeled off the skin, but that skin that you've peeled off was the epidermis. So now you kind of have a visual of what the epidermis is, because if you've ever had a blister, you've peeled it off. So what are some of the cells of the epidermis? Keratinocytes. It's a majority of the cells of the epidermis, so they fill up with keratin and die as they move toward the surface. Melanocytes produce the pigment melanin. There's different types of melanin, or skin pigment, that are produced, and the different concentrations of the different types of pigments gives everyone a unique skin color. Melanin production increases in response to exposure to UV light. I was talking about how DNA absorbs our UV light. Well, melanocytes are stimulated to produce more melanin in response to UV. And that's going to help us. The more melanin we have, the darker the skin, the more protected our underlying cells are from DNA damage. That's why when you have fair skin, you end up tanning in the summertime. Epidermal dendritic cells, or Langerhans cells, these eat substances that make their way into the epidermis. So if you ever do get a paper cut and have a bacterial infection there, these cells are going to help eat up that bacteria. And then tactile or Merkel cells, these cells provide sensory information from the epidermis. So they're light touch receptors. Light as in like gentle touch receptors. So they can feel tiny little touches on the surface of the skin. We have various layers of the epidermis. So if we go back to this picture to give you a visual, we are just talking about this part of the skin now. We're just talking about the epidermis. And of these layers, we start at the bottom and then we work our way toward the top. As we work our way up, the cells are dying as they're filling with keratin. So the bottommost layer is the stratum basale. The stratum basale, also known as the basal layer, is the layer that contains actively dividing cells. The process of cell division is called mitosis. So we say that this contains mitotic cells. So those are cells that are dividing or going through mitosis. Then we get up into the stratum spinosum. Now here you can really see the connections, those desmosomes that are anchoring those cells together. In a preserved tissue specimen, we have to dry it out in order to preserve it. So we do that with various salt solutions, and as we're drying it out, the inside of the cells kind of shrink, which causes these desmosomes to pull on the membrane, and that leads to the membrane looking spiny shaped. So that's how this layer got its name, really, is based on the rich desmosomes that make it appear spiny. Now right about here we get up into a different cell type, and you can actually see these different cell types. Here you can see all the desmosomes. Now here you can see a different region. This region contains numerous granules. So in the stratum granulosum, it contains numerous granules that are producing the protein keratin. So this is where keratin production skyrockets. And you can see those numerous granules in this layer. Then you get up to the stratum corneum. The stratum corneum is a majority of the thickness of the epidermis, and a majority of the cells here are dead. So it's basically 30 or so layers of dead sacs of keratin. So again, that stratum basale, it's a single row of stem cells. It's also called the stratum germinativum, but in this class we'll just call it the stratum basale. The journey from the bottom to the top takes 25 to 45 days. So every month or so, the skin cells are completely replaced from the surface. Now every day, these skin cells are sloughing off, and they're continuously having this happen. But if we want to track one new cell from the bottom to the top, it takes about a month or a little over a month. Now this stratum lucidum is only found in thick skin. So that would be the palms of the hands, and the sole of the feet. And the stratum lucidum is an additional layer that lies between the stratum granulosum and the stratum corneum. The stratum lucidum is not why it's thicker in these regions. It's just an extra barrier of protection, because if you think about an evolutionary standpoint, we used to not have shoes, so we used to walk barefoot around. And in order to prevent penetration of harmful substances through our hands or through the bottom of our feet, we had to have this extra physical barrier to prevent chemicals from getting through. So this is just 1 to 2 cell layers thick, but it really serves the role of preventing substances from getting through it, so that it provides extra protection to those areas of our body. Friction ridges. Friction ridges are fingerprints. They're the shallow lines on hands and feet. They're epidermal ridges on top of papillary ridges of the dermis. So these are folds of the epidermis that you can see. You can also see the openings of the sweat glands. So when the police do fingerprinting, they're not only looking at the lines that you see here, they're also looking at the arrangement of the sweat glands, and that's really how they end up matching up fingerprints. The dermoepidermal junction is just the space between the dermis and the epidermis. And this is where I talked about that blister, so you can see how pink it is underneath, and how this layer of skin can be completely peeled off and it doesn't bleed. And that's because this is the epidermis, this is the dermis. So now we get down to the dermis. And if we go back to looking at the whole skin layer here, now we're talking about this part, majority of the thickness of the skin. And you can see the 2 layers of the dermis, the papillary layer and the reticular layer. So this papillary layer follows right underneath the epidermis, and it gives blood supply to the epidermis. Since there's no blood vessels anywhere in the epidermis, the nutrients diffuse through from this papillary layer into the bottom part of this epidermis. And if you think about it, only the bottom cells are living. Only the bottom ones are replicating and migrating toward the surface. So the surface of the skin doesn't need nutrients, the bottom definitely does, and it gets it from this papillary layer. So the papillary layer contains capillary loops, and Messner's corpuscles, which also sense gentle touch, and free nerve endings, which sense pain. And the papillary layer nourishes the epidermis. Then we have the reticular layer. And with the reticular layer, this is about 80% of the thickness of the dermis. Contains collagen fibers to provide strength and resiliency, and elastic fibers to provide the stretch recoil properties of the skin. The reticular layer is where you're going to find a majority of the accessory organs of the skin. Cells in the dermis include fibroblasts. We just talked about those when we went through tissues, so those make up connective tissue. Since our dermis is made up of dense, irregular connective tissue, it makes sense that we'd have fibroblasts there. We also have macrophages present. Macrophages are going to eat debris that get into the skin, and that's where I gave the example of the tattoo. And then occasionally mast cells. Mast cells can cause allergic skin reactions like hives. So if you have a contact allergy where you touch something like latex and you break out in hives all over your body, that's caused by mast cells. And then other white blood cells may occasionally make their way there depending on if there's something going on with some type of infection. So here we go. The papillary layer again has those capillary loops, and then the reticular layer has a majority of the accessory organs of the skin. Deep in the dermis, we have these cleavage lines. Collagen fibers from the reticular layer of the dermis are arranged in bundles to form cleavage or tension lines. And this is where the cleavage lines are located roughly in the body. And for everybody it's slightly different, but it pretty much follows this same type of pattern. Incisions made parallel to cleavage lines heal more readily. So if you cut along a cleavage line, the stitches will hold a little bit better. If you cut across the cleavage lines, the stitches are going to be bulging out and the skin's not going to heal as well. So surgeons keep this in mind when they're making incisions through the skin so that they can cut appropriately to help with the healing after surgery. Flexure lines are these deep folds in the palms of the hands, any place that you have a joint where it forms these deep folds. Those are called flexure lines. So they're dermal folds at joints, and they're the areas where the dermis is tightly fused to underlying connective tissue. So know the difference between friction ridges, cleavage lines, and flexure lines. So friction ridges are the fingerprints, and they're formed in the epidermis. Cleavage lines are those lines where surgeons should cut across, and those are formed in the bottom of the dermis, the reticular layer. And then we have flexure lines, which are these deep folds, any place that you have a joint, and that's formed between the connections of the bottom of the dermis and the underlying connective tissue in the subcutaneous layer. So now we're going to look at injections. If you give an injection that's intramuscular, intramuscular means inside of a muscle. So that means that you're going to go at a 90-degree angle and inject all the way down into the muscle. Intramuscular injections go into the bloodstream quickly as long as they're soluble substances or polar substances. Many protein-based injections can go right into the muscle tissue as such. So they're fast acting because the muscle tissue has a rich blood supply, but it's not as fast as if you were to go intravenously, so into a vein, but it still works pretty quickly. Some vaccines are given intramuscularly. Subcutaneous, you're going to go on a 45-degree angle into the skin and attempt to get into that subcutaneous layer or the layer underneath the skin. Any fat-soluble substances, any hormone injections, those types of things are going to go into the subcutaneous layer. Intradermal, you want to go at a 10 to 15 degree angle. That means you're going to go into the dermis layer of the skin. An intradermal injection is going to test for immune stimulation. So if you ever have the TB skin test, they give you an intradermal injection and then try to see if any of the T cells in the area are reactive. And then you come back and look at the results of the TB skin test. They inject a small portion called purified protein derivative, PPD, from Mycobacterium tuberculosis. And if your body responds, it's a type 4 hypersensitivity reaction. Those T cells are going to cause a local reaction, and then you'll see a bulge of the skin coming up, and it'll have a color to it, and then you know that the patient has been exposed to tuberculosis. So those are just some examples of some types of injections.